Reagan. So today I want to teach you about the biology IA. Okay, so we're going to focus on the design part of the biology IA. Okay, where you have to design your own experiment and write out the hypothesis, the method, and all that. Okay, so online there are a lot of resources that probably teach you how to write biology IAs, but this video is truly unique because what we're going to do here today, not only am I going to teach you how to get a level 7, I'm also going to give you a very concrete example. Okay, I'm going to tell you very concretely an example of how you can do the biology IA. Okay, and so it will really paint a clear picture for you. Again, immediately after watching this video, you should be able to write your own IA. You can base on my experiment and then make some slight tweaks, and then you can have your own design lab. Okay, so me myself, I'm the co-founder of HKExcel. I don't normally teach biology. I've taken it in the IB, but actually in HKExcel every week we tutor over 1,000 students every week, all around the globe, through online and through live lessons. Okay, so the point here is that I've consulted with the biology experts at our company, and to make this video for you. Okay, so now let's get straight to it. Okay, so for your design IA, okay, there are a few components here. Okay, so the first step you start up with is the aim. Okay, the aim of the experiment. So as I mentioned just now, I'm going to give you a concrete example. The example here is very easy. Okay, it's what one of our team members did when he was in IB, got a level 7. Okay, so to investigate. So the aim for the design lab, as an example I'll give you, is this: to investigate how concentration of degradation enzyme, okay, affect the rate of liver tissue breakdown. So if you take biology, or even if you take chemistry, you know that enzymes speed up reactions, right? So basically you're asking, how does the concentration of enzyme affect the rate of reaction in which the liver tissue breaks down? Okay, so for the aim, you can write something like that. Okay, then for the hypothesis. Okay, for hypothesis, you need to give what you expect would happen. Okay, so for this particular example, of course, you can say the hypothesis is increased concentration will increase the rate of breakdown, right? That's the scientific theory, right? We all know that. But the point here is why. You also need to use scientific theory to explain why this is the case. Okay, so I would suggest, so the theory here is that the enzymes, so do you remember what enzymes do? Enzymes basically provide an alternative reaction pathway, right? That lowers if you take chemistry, it lowers the activation energy and speeds up the reaction. Okay, so the key point here is enzymes provide an alternative reaction pathway. Okay, after that one, so this to reduce activation energy. So this is a scientific term, activation energy of a reaction. The lower the activation energy, the higher the rate of reaction. So hence, increased rate of reaction. Okay, so this is just your hypothesis and the theory to back up why this is the case. Okay, next, you need to talk about the variables. Okay, what are the variables in your experiment? Okay, so if you're a decent science student, you should already know this. Okay, what does independent variable mean? Well, independent variable is the variable where the scientist is actively changing, right? So what the scientist makes an effort to change, that is the independent variable. And then what is affected is the dependent variable, right? So what are we trying to change here when we are doing the experiment? Which variable are we proactively changing? So it is basically the concentration of degradation enzyme, right? The concentration of degradation enzyme. But one thing you want to take note of is that in the dependent variable, you should also state clearly what are the concentrations you will test. Okay, so like when you're testing different concentration, which different concentration? So you can, for example, say different concentration to be tested are, so for example, so you have 0 molar, so 0 moles per milliliter, or 0.1 molar, 0.2 molar, 0.3, 0.4. So the point here is you need to at least have 5. At least have 5. Yeah. So for independent variable, at least test 5 different numbers. Okay, for independent variable. Okay. Now, the dependent variable. Well, so the dependent variable, what is the dependent variable here? So what is going to be affected by the concentration of enzyme is the rate of change, rate of liver tissue breakdown, right? That's obvious, right? Now, let's go to the control variables. So because when you're doing an experiment, you're looking for causal relationship, right? Cause and effect. So you want to keep all the other variables constant, okay? So that we can only see the effect of the changing the independent variable. If every variable is changing, you don't know what variable is causing that change, right? So what are some important variables to control in this experiment? Well, so the way you can think about it is what other factors affect the rate of reaction? Okay, we want to keep all other factors the same and only change the concentration. So what other factors affect concentration? So of course, temperature, right? Temperature. The temperature is definitely, if you take chemistry, you know, temperature is going to affect the rate of reaction. The second thing that affects rate of reaction is basically the surface area, okay, of the liver tissue. So if you study chemistry, okay, if the surface area of liver tissue is bigger, let's say you have a liver tissue that is acute versus a liver tissue that is just like basically in powdered form. So in powdered form, there's much more surface area for reaction. Actually, it speeds up the reaction dramatically. Okay, and of course, the third one is the enzyme volume. Okay. So, like if you have a higher volume, you have more enzymes there, and cause it to be faster. And in a controlled variable, you want to explain how you keep it constant. So for example, for temperature, you can just say, how do we keep temperature constant? We keep temperature constant by performing the experiment in the same room, okay, setting the air conditioner at the same temperature. Okay? The surface area of liver tissue, so you can, for example, say we will cut liver tissue into 1 cm by 1 cm cubes, okay, for example. Enzyme volume, so for example, you can say the enzyme volume is going to be kept at 50 milliliter. Okay, it will be measured carefully by a pipette, right? Something like that. Okay. Okay, now so let's go to the method. So for the method, you basically just need to chronologically talk about what are the steps in doing the experiment. Okay, so let me give you a quick explanation of how this experiment is done. Okay, I'll tell you verbally how this experiment is done, and then I'll tell you how to write it up. Okay, so this experiment is done basically. First of all, you need to have liver tissue cubes. Okay, and then you put it in the enzyme solution. Okay, when you put in the enzyme solution, actually, gas starts to evolve. Okay, because of this, that's the nature of the reaction. Gas evolve. The gas revolve, and this tube here is what we call a burette. Okay, and you attach this burette. So this burette, all the gas that is being evolved will go through this tube because you have the burette which prevents the gas from escaping. And as the burette goes into it will eventually flow into the syringe. So for the syringe, as gas goes into the syringe expands. This is going to go, this handle is going to leave, and you can see how much gas is being filled into the syringe as more and more gas goes into the syringe. Okay, so of course during this experiment you need a timer. Okay, so maybe once you put the liver tissue in, you can put the syringe on put a burette and the syringe on, and then you start a timer. Okay, so the timer is, let's say, you time it for 1 minute. Okay, so after 1 minute, you see how much volume is being put into the syringe. If a lot of gas is being transported into the syringe, it means that the rate is very fast, right? If not a lot of gas is being transported into the syringe, it means that the very little gas is evolved. The rate is very slow, right? So that's pretty much it. Okay. So now let me teach you how to write the steps. Okay, exactly what are the steps in your write-up? Okay, so in your lab, like in your method. So first, you want to talk about how you prepare 5 different solutions with different concentration. Okay, I'm going to be a brief here, and write it up. Just pay more attention to detail. Well, 5 different enzyme solution. Okay, you need to explain how you prepare. So by using a standard, maybe one molar solution, okay, very concentrated solution, and performing dilution. Okay, I don't think you need to explain how the dilution is done. Just say that. Okay. Next step. So you can also say that. Okay. Then your second step. Of course, now we need the liver tissues, right? So you can say cut liver tissue into 1 cm by 1 cm cubes. Okay, right? So using a ruler to measure. Okay, using ruler to measure. So because you need to ensure that every single cube has the same size. Okay. So and then say next step, of course, is place the enzyme solution into beaker, right? Obvious. And then what is the fourth step? Place the liver tissue into the solution. Okay. The fifth step. Okay. Then you can say something like this. After 10 seconds. Okay. After 10 seconds, with a timer to ensure. Okay. So after 10 seconds, you have to you place the liver in the beaker. You place the liver in the beaker, and then you put the burette, right? So this you put the burette on. Okay. So place burette with the syringe. Okay, onto the beaker. Okay, right? And then you can just say after 1 minute you measure the change in volume. Measure how much gas is put into the burette, right? So I'll record volume of gas that went into the syringe. Okay, measure how the volume of gas that went into the syringe. Okay, for this, actually, one of the main things that the IB asks you to do is that you need to record sufficient data. You need to record sufficient data. Okay, so normally we like to say that it's called the 5x3 rule. So what you want to do is have 5 different variables, 5 different independent variables. So let's say we're looking at the concentration. We have to look at 5 different concentrations and 3 trials. Okay, so at the end you can say repeat steps 1 to 6 use for 3 times using the enzyme solution of the same concentration. Okay, you can write that down like step 7, right? Just repeat using the same concentration of enzyme solution for 3 times. So that's how you get 3 trials. And then at the last step, you can say repeat steps 1 to 7, okay, using enzyme solutions of different concentrations, which includes 0.1, 0.2, and so forth. Right? So this will allow you to collect enough information. Okay, you have 5 free trials and 5 independent variables. This is very important. Another thing to look out for is that for the variables, you want to make it explicit that you're controlling the variables. Maybe actually in the first step you can say, in the last step you can write something like ensure that all steps above are performed in the same room under room temperature, right? Or for example, And then here I want to make it clear it's using a ruler so that the liver tissue size is the same. Okay. So of course, now it's your turn. I think now you should have been enlightened, right? You probably know how to do the biology design IA. Of course, I would highly recommend to do something related to enzymes. It's very easy, right? You can just follow this video, just change some parts, right? So you can say how enzyme affects the rate of, for example, muscle tissue disintegration, something like that, right? So until next time, I hope you find this video helpful. If you want to have a biology IA sample, you can go into the link below and basically fill in the form and you can get the sample. Okay.